For 30 years, Alzheimer's research has been divided by two competing etiologies. On one side, the amyloid cascade hypothesis places parenchymal amyloid beta at the origin of the disease. On the other, the cerebrovascular hypothesis argues that blood-brain barrier breakdown and microvascular hypoperfusion precede the parenchymal injury. This schism is institutionalized in the current ATN clinical diagnostic framework. By defining the disease strictly through amyloid, tau, and neurodegeneration, the framework treats vascular pathology as an external comorbidity rather than an integrated driver of the disease itself. Yet the autopsy data contradicts a pure parenchymal model. Epidemiological studies of community-dwelling older persons show that mixed pathology, the co-occurrence of cortical infarcts, small vessel disease, and classic Alzheimer's markers, is the modal finding. Pure Alzheimer's pathology is the exception in the aging population. The failure to reconcile these two frameworks creates a structural impediment to clinical trial design. Patients with dominant vascular pathology are misallocated in trials targeting only parenchymal markers, directly confounding the results of targeted interventions. The ATN model is structurally incomplete. It measures the damage strictly within the brain, ignoring the vascular interface, where systemic aging signals are first transduced into the central nervous system. Late-onset Alzheimer's is a stereotyped tripartite temporal cascade. The vascular and parenchymal substrates engage in a precise, phase-specific sequence across the adult lifespan, mapping onto a single chronological axis. The cerebrovasculature executes a distinct role at each phase, conditioning the initial failure in the brainstem, gating the inflammatory transition in the hippocampus, and amplifying the final structural collapse in the cortex. The molecular spine uniting this sequence is vascular cell adhesion molecule 1, or VCAM1, expressed on the brain endothelium. It acts as the physical gateway between the aged systemic blood supply and the protected parenchymal compartment of the brain. This four-substrate account integrates disparate clinical data, showing how the cumulative load of midlife hypertension and vascular stiffening accelerates the failure of the brain's primary barrier. The sequence begins in the 3rd to 5th decades of life. The disease's earliest parenchymal target is the locus ceruleus, a densely packed noradrenergic nucleus in the brainstem. The locus ceruleus is unusually vulnerable due to its vascular architecture. It is metabolically hyperactive, yet relies on a single arterial source. Arterial spin labeling MRI reveals midlife perfusion decline. Long before cognitive symptoms appear, it experiences recurrent subischemic intervals. This hypoperfusion restricts oxygen availability at the exact moment the cell requires it. Without sufficient oxygen, the locus ceruleus neuron loses its capacity to manage the natural auto-oxidation chemistry of noradrenaline, generating a severe spike in mitochondrial oxidative stress. When this stress crosses a critical threshold, it triggers sustained PARP1 hyperactivation. The cell rapidly exhausts its NAD+ reserves, compromising sirtuin signaling and mitochondrial efficiency, culminating in local bioenergetic failure. The midlife vascular decline is the upstream conditioning event that dictates the exact timing of this bioenergetic ignition. As we move into the 5th through 7th decades, the pathology targets the hippocampal temporal axis. The hippocampus has a constitutively leakier vascular architecture, marked by lower expression of tight junction proteins like claudin-5, compared to the cortex. This structural vulnerability intersects with the Youssef-Weiss-Correa mechanism. Over the human lifespan, the concentration of soluble VCAM1 in the systemic plasma increases approximately 5-fold. This aged systemic blood exerts continuous pressure, inducing dense VCAM1 receptors on the hippocampal endothelium. Aged monocytes bind to these receptors, activating NADPH oxidase and sending a concentrated pulse of reactive oxygen species directly into the endothelial cell. The oxidative pulse activates the NF-kappa B transcription factor, forcing the endothelial cell to secrete inflammatory cytokines, specifically IL-6 and CCL2, across the blood-brain barrier and into the abluminal parenchyma. These cytokines diffuse into the surrounding tissue and reach the resident microglia. The sudden inflammatory load degrades the homeostatic microglial signature, suppressing protective markers like P2YR12, and forces the cells into a chronic, disease-associated state. Hippocampal microglia lose their homeostatic state because they are responding to specific, age-upregulated inflammatory signals gated by the brain endothelium. Entering the 7th decade, the pathology expands into the cortex. The clinical presentation of dementia emerges as the cortical microcircuit loses its gamma-frequency computational drive. The parenchymal executor of this cognitive decline is microglial MMP9. This secreted metalloproteinase digests the perineuronal net, the dense matrix ensheathing parvalbumin-positive fast-spiking interneurons, stripping away their metabolic and structural support. This synaptic failure has an exact vascular analog, the blood-brain barrier's basement membrane. Microglial MMP9 digests both matrices simultaneously. As the perineuronal net dissolves to disintegrate the synaptic circuit, the adjacent vascular basement membrane fragments, destroying the barrier. This barrier collapse is amplified by cerebral amyloid angiopathy. Soluble amyloid beta 40 cleared from the brain deposits along the fragmented basement membrane. It systematically replaces the vascular smooth muscle cells, obliterating the vessel's ability to regulate blood flow. Phase 3 is driven by a singular mechanism of matrix metalloproteinase overactivity. It executes the structural disintegration of the brain by destroying both the vascular and synaptic compartments at the same time. This late-stage vascular degradation explains the adverse effects of modern anti-amyloid monoclonal antibody therapies. When these drugs recruit microglia to clear amyloid from already compromised vessel walls, the structural integrity of the capillary fails, resulting in plasma leaks known as area E and microhemorrhages known as area H. Area is a mechanical failure of the vascular substrate. Clearing amyloid from a vessel wall that has already lost its smooth muscle layer inevitably compromises the structural integrity of the capillary. To account for this, the field must adopt an ATNV diagnostic framework, adding a precise vascular axis. This requires tracking Phase 1 cardiovascular profiles and regional perfusion, Phase 2 soluble VCAM1 and PDGFR beta levels, and Phase 3 cerebral amyloid angiopathy imaging. Relying solely on parenchymal markers guarantees the misallocation of subjects in clinical trials. It attempts to measure the damage, ignoring the temporal pacemaker driving the progression of the disease. A complete model allows for a developmental pharmacology of Alzheimer's. This begins with rigorous midlife cardiovascular control in Phase 1, progresses to VCAM1 antibody blockade and parasite stabilization in Phase 2, and concludes with strictly area-stratified anti-amyloid clearance in Phase 3. The disease-modifying effect that has evaded researchers for decades will not come from a single molecule. It requires the cumulative impact of targeted interventions across all 4 biological substrates as they sequence across the lifespan. Oscar Fisher's 1907 recognition identified the inflammatory and vascular response as a central driver of neurodegeneration. Today's molecular biology confirms Fisher's century-old insight. The vascular phasing of late-onset Alzheimer's disease offers a cohesive temporal architecture, proving that the cerebrovascular interface is the primary gateway through which systemic aging dictates cognitive collapse.